All right, welcome everyone. Good morning in my time zone in Ottawa. Good afternoon in Europe, and good evening if you're in other parts of the world, in Asia, for example. I'm Paul White. I'm a research scientist at Health Canada, and welcome to the second day of our course on use of the benchmark dose approach for quantitative analysis and interpretation of genetic toxicity dose response data. I'm just going to go over the program for today in a minute or two. Today we're going to have examples, real-time examples of data analysis using ProstWeb. And you can see there the first part is going to be on continuous data. Then we'll have a short question and discussion period. Then the second part on quantal data. Guangchao Chen will show how to analyze continuous data. And then George Johnson will show examples for quantal data. Then we'll have a short question and discussion period, then a break, and then open discussion and questions for anything about the course. So day one or day two content, and that will be 45 minutes. And we'll end a bit earlier today. If people are still available, we could continue, but we certainly wouldn't go beyond 10 o'clock. So getting right into it, real-time dose response analysis using ProstWeb. You can see the web address there. I'm going to talk for a few minutes about how you need to prepare the data, and then I'll pass it to Guangchao, who will show examples of real-time examples using ProstWeb to do the actual data analysis. So I just wanted to say a few words of the data sets that we've selected for the analysis. So we've scoured the literature. I've tried to look at the recent literature for examples that are, I guess you could say, contemporary. For example, about comparison of nitrosamine potency. We have 3 data sets that Guangchao is going to cover, and then we have 2 alternative data sets that he may cover if time is permitting. The first one is about peripheral blood micronucleus assay with mutamouse males exposed to benzopyrene. That's the mega BAP experiment that I talked about yesterday. It's a 28-day oral gavage experiment with 2-day sampling time, 10 doses plus control, a minimum of 7 animals per dose group, and that's individual animal data. So in that case, we have replicate animal data rather than summary data, which would be the average and standard deviation, for example, of the response for each individual dose group. The second example is a mutant frequency assay in mutamouse. So mutamouse males exposed to NDMA or NDBZA, so nitrosodimethylamine or nitrosodibenzylamine. I talked about this one yesterday, and Guangchao will walk through how to do the analyses. In this case, it's a single oral dose, and generally use either a 30 or a 90-day sampling time, and its liver is the tissue. And Guangchao will show how to analyze just the 30-day sampling time data. It's 3 doses plus control with 5 animals per dose group. And in this case, it's summary data. So it's not actual individual animal data. The third one is mutant frequency assays. And in this case, the target locus is either GPT or C2. And I talked about this one yesterday too. It's a comparison of big blue mouse, big blue rat, GPT delta Fisher 344 rat, and GPT delta Sprague-Dawley rat. And in this case, the exposure is to NDEA, nitrosodiethylamine, at 28-day repeat dose, and the sampling time is 0 to 3 days. So it's variable across the studies. And the dosing, the treatment regime is oral gavage or drinking water. The GPT delta is drinking water. The others are oral gavage. Liver, and you can see here, I won't read all of this, but different numbers of doses and different numbers of animals per dose group. Again, this is summary data, and the data come from Burku et al., Zhang et al., and Akagi et al. So we've collected these, in that case, we collected the data very recently, specifically for this course and for this example. If we have time, we also have C2 mutant frequency data in big blue male rats exposed to either NDEA, nitrosodiethylamine, nitrosapyridine, or NPIP, and you can see the full name of NPIP down below. Sorry, not NPIP, sorry, NTTP, and here's the full name of NTTP down below. And in this case, it's 28-day repeat dose oral with 3-day sampling time in liver, and you can see here the number of dose groups and number of animals per dose group. Again, this is summary data with very recent data from Burku et al. And Pauli et al. And last but not least is data from 2 of Ali Long's publications. It's LAC-Z mutant frequency, mutamouse males exposed to either benzoapyrene, dibenzo-H-anthracene, benzo-B-fluoranthene, or benzo-K-fluoranthene. And this is 28-day repeat dose with a 3-day sampling time. We're only going to talk about bone marrow. And as I mentioned, at least for benzopyrene, it's many doses, 10 doses plus control, fewer doses for the other compounds, and 4 to 7 animals per dose group. In this case, it's also individual animal data. So those are the 5 data sets that we've collected for the purpose of real-time demonstration, and we'll see how much time we have to get through these various data sets. So just a few words about initial data processing. Whether it's continuous or quantile data, it's pretty much the same. So you need to make sure, in this case, we've prepared data files in Excel, that you have all required variables. And of course, that will vary depending on your research question. So you need a variable for the covariate, for example, if you're comparing across whatever it is, transgenic rodent assay variants or chemical or whatever it is. And you need appropriate column headings. So there's guidance in the PROST manual, specifically the annex to the PROST manual, about things you need to consider. So for example, don't use really long column headings because it'll interfere with the visual output of the graphs, for example. Don't use special characters, and it's best not to use spaces. So we use either underscore or dot. Save as tab delimited text only file. That's fairly straightforward, and I'll show an example just very briefly. You need to import the data either into PROST Web Online or in the R version of PROST. Of course, you could use those other tools that we mentioned yesterday. You need to select the required portions of the data set if necessary. So I mentioned in the Zhao et al. Paper, there's both 30 and 90-day sampling time, tissue sampling time. So you may only want to use one of those. So you may want to select 30 days and 90 days in a separate analysis. Select dose and response columns, very straightforward. Indicate the data type, either continuous or quantile. Select columns containing, for example, standard deviation, standard error of the mean, and group size where applicable if it's summary data. You don't need to do that if it's per animal data, replicate data. Indicate the benchmark response. Select the covariate if applicable, and select model averaging if desired. And Guangchao is going to go through all of this so you'll see exactly how it's done. And as I mentioned just a moment ago, you can look at the PROST manual, then specifically Annex 1 for guidance on data formatting. So I'll just jump out of this and switch over to Excel. So in this case, the file name, this is for the mega BAP experiment, and these are the micronucleus, the in vivo micronucleus data, and it's very straightforward. I mean, I just have animal, chemical, it's all benzopyrene, dose, percent micronucleus for reticulocytes, and percent micronucleus for mature red blood cells. And here's the endpoint micronucleus. Don't necessarily need that variable, but you can see on the bottom here, I have worksheets for both micronucleus and PIG-A. So those could be analyzed separately. And you can simply make a text-only file. It's very straightforward, and I think most people know how to do this. I would just do save a copy, and then it would be tab delimited text, and I can just call it whatever. I mean, I can call it test for these purposes here, and just save it, and that's it. And it'll give you a warning saying that it can only save one worksheet for it in this format. So it'll save one worksheet at a time. So you just say, okay, and now it's been saved. And I can just switch over here using Notepad. I can see what that looks like. So here's the file name with the .txt ending. And you can see here this is the animal specific data from that Excel file as tab delimited text. So very straightforward. And then that gets imported into Excel, into PROST, almost said Excel. That gets imported into the web version of PROST. So I'll just leave it there, and I'm going to stop sharing, and then I'm going to pass it to Guangchao, who's going to work through these examples using the online version of PROST.
Yeah. Okay. Thank you, Paul, for the lovely introductions. I'm Guangchao Chen from RIVM, the Netherlands. It's nice to see you all. I'll be sharing my screen first. Let me try to find the right one. I hope everyone sees my screen now with the web page. So today we will walk through a few examples of BMD analysis with the Web2 PROST web, and to see how things are done in reality and what are the things we need to look into and how the analysis actually look like from the output of this tool. To do that, we firstly need to go to the web page of ProsWeb, which is here, prosweb.rvm.nl. And then if you actually copy paste it, come here. Oh, actually, there'll be a pop-up, something you need to click OK, but I guess I already did that. But that's not very important here. Then here we see some information overview, which is the analysis you have done, since we have done nothing yet, so there's nothing. But then you have some columns, analysis, name, status, created on, expires. I will explain later when we did some analysis together. For now, if you want to do your new analysis, you need to click create new analysis, and then this is the page we see then. So firstly, let's just briefly take a look at the tool first, then we begin with the examples. But don't worry if you don't remember all the details, because the introduction also in the manual version of the ProsWeb. So the manual start is online. Everyone can find it. So don't worry about that. You can come back to, to the manual all the time. So basically what we're looking at now, we start with, so here, analysis. So some region here, you can upload your data and choose some settings. And here is example data set, which means you can also use some example data set that's already inside here, if you just want to check how the tool works and stuff. And then you will give it a name. Of course, you need to select what you want to do. Here we do those response analysis, and then we can continue. So this is, in general, what we are looking at here. Then let's start with some concrete steps. The first thing is to upload the data. Paul already mentioned that the format of the dataset is tab delimited. So that's a TXT file. So let's choose the file for example 1. The BPA micronuclears. After choose the file, we have to click upload. If you don't click it, the dataset is not uploaded in the tool. After you click it, you can find in the bottom of the web page the uploaded data. Take a look what are there. Always look at your data first before your analysis. For instance, what kind of information do we have? Here we have the animal number, different dose groups, quite a few actually, and then 2 different endpoints, and then it just said what endpoint is. So it's a rather simple data set in this case. So that's all good. Let's continue further. The next thing we need to do is give this one a name before you continue. So here we can say example 1, example A. Let's do that. And click specify. And here we have a new page. In this page, you can specify the more detailed settings. For instance, which is the dose, which is the response, kind of type of data it is. So let's just go check that step by step. Firstly, the dose column here is quite obvious. We have the dose. The response column, we choose this response for the example. And in this case, each animal has their own response value. And in this case, we call it individual continuous, or continuous value for each individual. That's pretty direct. And then we go further down. So there's no other information, covariate, BMR. So BMR is very important. We say, okay, for in vivo genotoxicity, let's just use 50%. And we just click run. And this is rather simple example. I think it will finish pretty quick. So we will just wait until finish it. And then meanwhile, now we see that there are some extra information here underneath. Oh, that's pretty quick. So the first thing is analysis name. That's the name you just specified. And this is status. I think the first one will be initiated. And then afterwards, it will be proceeding or something like that. I forgot. We will take a look again. And then when the analysis is done, it got finished. So that means you will get your results. And then here you can see what time your analysis is finished, and then in principle after 24 hours the record will be removed from this web page. So you have 24 hours to go back to your old analysis, or just copy paste it and to save it on your own folder. And then we have a few buttons here. The last one probably quite direct. It's just delete. If you don't want it anymore, just remove it. And then if you put the cursor on the symbol, then you can see what that means. So the first called specify, second is detailed analysis, third is report analysis results, and the last one, advanced plotting. So the first one is in the situation that you want to actually change the setting of your analysis. For instance, maybe I choose the wrong information, maybe I use the wrong BMR, then you click it, you adjust it, and you run again. But then this new analysis will overrun the old record. So you only find one record. So that's the first function, specify. The second one is details of the analysis, which look like this. So besides the input, and then also print some output, which is for a lot of users, it may be less interesting, but it does just give you some more detailed information about the console in outputs. What's the most important thing is actually this part, the report analysis results. So this is what you should look at if you're mostly interested in the results of your BMD analysis. And then in this page, we start with the name. So that's the name, still the name you gave. And then this is the time you created this analysis. Meanwhile, it's the version of the tool you're using. And then the first section of this page will be input values. So those are all the things you specified. For instance, whether you removed any data. In this case, we did not. In other examples we made, then you will see there's some extra information here. Also, the type of response data here we say is individual continuous. So we have the response value for each individual, and it's also continuous endpoint. And we choose which column is the dose, which column in the dataset the response. We did not use any covariate here and other things. And the BMR we selected, and model averaging we did not use in this case. And then the output of the analysis, the first output will be the individual model fit. So in this case, the model output two individual model fit, one for the exponential model, one for the Hill model. And then again, like yesterday we mentioned, you should look at firstly the individual model fit, just to have a feeling. How is the model describing my data? Yeah. So here we can see, okay, more or less it looks normal. So nothing odd popped up. And meanwhile, there are also some other information, for instance, for each individual model, the estimated parameter. You still have, I'm not sure if it's big enough for many people. So you still have the version, the log likelihood, which shows how good the model fit is. The AIC index, which also indicate the model fit, but also taking into consideration number of parameters. So the AIC actually determines the weight of the models in the case of model averaging. But in this case, it's just another index to show how your model fit is. And then you also have the value of the parameter, for instance, the variance of this analysis. Also the parameter A, which is the background estimate, and B, and here we have the CED, which is the potency, which is around here. We have this parameter C, which is maximum fold change. So here we see the increase is around 4.6 fold. And then we have D, which is the shape parameter. The D is basically how steep this curve is. So those are the basic parameter of the models. And then afterwards, you have the output around the CED and also which BMR you used. Like yesterday we said, for continuous data, the BMR, the benchmark response, is also termed as critical effect size, but they are actually the same thing. It's the benchmark response. Here we have the lower bound or the BMD and the upper bound of the BMD. I will make it smaller. So after looking at the model fit of the curves, here are some other output information. It will show you how well each model fit is in terms of log likelihood, also number of parameters of each model, and also the AIC index. So the AIC, like I said, it also reflects how well the model describes the data, but then it takes the number of parameters into consideration. Because in principle or in general, when you increase the number of parameters, then the model should actually describe the data better in some case, but then it could lead to the situation of overfitting. So actually this index, AIC index, it penalizes the adding of parameters. So this is the whole idea. And the last bit is your BMD confidence interval. You have the lower bound and you have the upper bound. So here the BMD confidence interval is quite narrow. So it's the factor less than 10, and then which is quite good, I would say. And this, if you do risk assessment, this would be one of the point of departure you could consider. So this is the general output information for this example. And we have actually one more button here, advanced plotting. If you clicked it, you see here, for instance, for which model do I want to plot? You can adjust. So here, default both models, but you can select one of them. And then here you have other settings you can change. Do I want a log or original scale or square root? I wouldn't recommend to do that, but those are the options here. And you can also adjust the limit of your plot, yeah, for X, for Y. And also you can adjust the size of the data point, the mean. And there are some other options you can change, maybe the titles for the X and the Y, and even the color. Yes. So those are the options, but I think it's less travel for many people. That's everything for the first example, quite simple and straightforward. And then I think we can continue with the second one. Let's go back. The second example is multi-mouse, yeah, multi-frequency. Let's go to that data set. Again, choose file, B, open, and I need to click upload. By the way, one thing I forgot to mention is you need to select the correct decimal separator, because I think in some countries people use comma as decimal separator. But in this data file, it's point. So you need to choose the correct one. Otherwise, you run into errors in your analysis. It's small details, but it did happen before. People can't get anything they want just because it's small thing. Uploaded the data, and let's take a look at the data again. See what we have. So we have information on the chemicals. So we have two chemicals here. The string of the animal, mouse, which is just one. And then we have the TGR, what kind of tissue, as well as the exposure route, so application time, fixation time, and we have the dose and response. And in this case, we can see that the dose response data is reported per dose group. So for instance, for dose group 2, and there are 5 animals, then the mean of the response is 84, and the standard deviation is 18. So this type of data, we call it summary continuous data. So it's still continuous response, but then the data is reported as a summary for each group. Okay. Then we continue with our analysis. I think for this data set, we choose 30-day fixation time. So here is how you can select your data. You just click this downward arrow here, and we select 30. So we just unclick 90, right? And that's it. And you can also, of course, do other selections by click or unclick. So it's rather straightforward, I think. Let's go back. we give this analysis a name again. Let's say example B. Dose response, specify, come back to the same page. Let's choose the dose, which is the dose. And let's choose the response, which is also response. So here we said it's continuous but summary data, so we choose this. And in this case, you also need to tell the tool which column is the dispersion measure, which is standard deviation. But then, of course, you need to select this is standard deviation, not the standard error of the mean. And then, meanwhile, you need to tell the tool which is the group size, which is group size. And in this example, we will use chemical as covariate, which we have two here. And let's again just always use 50% BMR in our analysis. Okay, so in this situation, we will also use model averaging. Then it's simple, just click. Because this is for a demonstration purposes, so we will just leave the number of bootstraps as default 200. And that's it. We click run. Oh, okay. So now you can see the beginning is called queued. So queued means the analysis is not assigned to the server of the tool yet. And now it's called in progress, which means the calculation, the analysis is ongoing. So that's the status. And the last one will be finished. I think because this one has model averaging, it will take a bit. So let's not just waste time to waiting for it. And yesterday I did exactly the same analysis for each examples. So let's pretend right now it's after 1 hour already, and this example B is already finished. And let's just jump onto that, which is this one, this row. So again, all the information the same as explained. The only difference is the output. So we go to the output. Again, the name, the time, and the input values. Here you can see remove data. Yes, we did. Which data we removed? We removed 90 days fixation time. And the rest of also just the settings of your analysis. And then here we have the graphical output. So here we chose two chemicals and use the chemical as covariate. So of course, then we see firstly the results of the model averaging for two different chemicals. And then for each chemical, there's also a BMD confidence interval based on model averaging. And then the output is here, like we also introduced yesterday. So those are different information of your model averaging analysis, version, also number of bootstrap runs, which BMR you chose, as well as the BMD confidence interval, which is also in the graphical output, but also down at the end of this web page. So here is also a picture, a plot of the BMD confidence interval, one for the NDMA, one for the NDBZA. Here we can see the confidence interval is quite narrow, which means the data quality is quite informative regarding the dose-response relationship around the BMR we chose. So here we can say, okay, NDMA is more potent than NDBZA. And I think it's also quite obvious if we look at this figure. And then we go down again. So here's the single model fit. Again, as we said, it's good, always good to look at the single model fit, just to have a feeling about, firstly, how does each model describe my data, and also just take a look at your own data, and then eventually you will have some accumulated feeling of how our good data feels like for the dose BMD analysis, right? So I think it's also quite important over time. And here we see actually the dose response shape is quite neat, and then that's also reflected in the BMD confidence interval. So when the data is less informative about dose response, your BMD confidence interval tend to be larger because it's more uncertain. Your BMD is more uncertain. And vice versa, when the data contains really good information on the dose response information, then your BMD is less uncertain. So that means the BMD confidence interval is narrower. And then we still have the output of the model fit. So here are different models being fitted. So obviously we have actually full model families, exponential, Hill, inverse exponential, and log normal model, full different model family being fitted. And then the best model of each model family is chosen for the model averaging. Here again, you have a log likelihood output, how many number of parameters, and you have the AIC. Those are just for the information. And then if you actually look, for instance, log likelihood for this, the value, the higher the log likelihood, the better the model fit. And for the AIC is the other way around. So the lower the AIC is, the better the model fit. So let's take an example, for instance. Here we see which one is the lowest. We have this one, the lowest. So that means exponential model 3 BV should be the best model fit. And if we come up, here. So we see indeed the exponential model 3 with BV is the best model fit got picked. So got picked. And the same goes to the other model family. So this is just for you to understand a bit better, what's going on in my analysis, and why. Yeah. And the last bit, second last bit, we have the model weight. So these are the information for the model averaging. So then you know actually how much weight of certain model a certain model has in the model averaging. And in this case, we can see actually they're quite even. So they are more or less evenly contributed to the model averaging. And that you can actually also see from the figure, more or less just out of feeling, they all describe the data quite well. So it also makes sense this way. And the last bit is just the output of your confidence interval of two different compounds. And those information also in the figure, of course. Yeah. Just for the convenience of saving your results, you can just simply select everything, and you can just copy paste it in a Word document and just save it. It's quite easy this way. Yeah, exactly. I think we're done here for the second example. Let's continue with the third, which is in the EA. We have 4 strains. Okay, good. Create new analysis, as always. In my data point is the decimal separator. Choose my file and the EA data, open, click upload. And we go down, look at the data first. We have a lot of information in this data set. The chemical, we have only NDEA, and we have strain. We have 4 strains, yeah? And then we have the information on the TGR, on the tissue, here's only liver, and the exposure route, here's garbage and water. We have application time, we have different doses, and we have the response. And here you can see already, so this is continuous response. Meanwhile, they are reported per dose group. So as we know, this is summary continuous data. Okay, so this is the data we have. And for this one, we're just going to continue our analysis. Firstly, give it a name. Let's say example 3. C. Then specify. Again, we come to the page of different settings. Let's choose those. Choose a response. Then we said is summary data. Then again, we have to choose which column is the ISD or ICE here. And here we have the standard deviation. We need to click. And then we need to choose also which column is the group size. Here, for this demonstration, we will use strain as covariate to compare the sensitivity between different strains. And let's, again, just use 50%, which is less important, but let's stick to the same number. And in this example, we will also use model averaging. click it and you see. Let's just use the default since this is just a demonstration, and we click run. Oh, actually, we saw the second one already finished, but okay. Here we have 4 groups. So again, my analysis is being progressed. So it's at in progress. I think we will, the analysis will take a bit, so we will not wait for it again. And let's just pretend after, for instance, 2 hours, our analysis is finished. And then we just jump right into that analysis, which is this row, actually. I give it the wrong name, but which is this row. Let's click. Again, go to this button, click. Yeah, it's the correct one. Name, time, analysis finished, input data. Here we can see string is covariate, and then we did use model averaging, and the number of bootstraps is 200. Perfect. Here is firstly the output of the model averaging. So we see that there are 4 different colors. So that means 4 different subgroups, and each subgroup relate to one strain. Again, here we have different information, the version, the BMR, and then we have the confidence interval. So all the information here as well. And then the figure below again is the plot of the confidence interval. And here we can see the BB rat and BB mouse, they are more or less equally sensitive, and the GPT rat and F344 and the GPT SD rat, they are more or less equally sensitive. And this BB is more sensitive than the GPT. So this is what we can see from this plot. When we go down, we again have the individual fit of the 4 models, and more or less we can feel, okay, the models describe the data pretty well, so nothing out of ordinary. Here as well, they all look quite okay. Again, we have on the right side, output information of each parameters. Like we said, parameter A here is the background response, which is the estimated background, by the way. We have the CED, which reflect the potency, and we have the parameter C, which is maximum fold change. So where it indicate where the dose response curve gonna level off, and we have the steepness to see how steep this curve is. And then we go further again, like we said, it shows you some detailed information of each model it fitted. Again, we have in model averaging, it's a standard. Like the 4 model families will be selected, and the best model fit from each model family will be chosen in the model averaging. Here again, we take another example. For instance, we go for this. We said for the AIC, the lower the better fit. So which one is the lowest? So I think it's the LN3AB. I think. So let's go check if it's the case. LN3AB. Yeah, it is LN3AB. So the best fit of the model from each model family is chosen for the model average. Again, we have the model fit. Here we can actually see the log normal model has, oh, sorry, wrong. The inverse exponential model has the most weight. That means it describes the data better than the other models, so it gets a bit higher weight. And then the exponential model got the least weight. That means compared to the others, this model describes the better, the least ideal. That's the case. And then in the end, we have the BMD confidence interval for the 4 strains or the 4 subgroups. So this is where you can find them. I think here are all the 3 examples, and I think we've done pretty quickly. There might be time to show the other. Paul, is there time to show the others?
Well, we're a bit over time, but I think we'll pick up time when we move on to the quantal examples, because
okay.
I think they're very quick. So I think we can afford to do one more example, and the question answer will be a bit late. If you want, we can look at the most common question just for the time being, before you go on to the next example. So the first question, which had the most votes, is in which cases should we use model averaging?
Well, also depends on the purpose of your BMD analysis. But if the intention is for a chemical risk assessment, model averaging is the standard way to do it now. So if you intend to calculate BMDL as a point of departure, always use model averaging. That's also in the guidance, the standard guidance. Yeah.
Okay, so let's go for one more example, and then we can get back to a couple of questions and then move on to the quantal data and examples. Oh, and one thing I keep forgetting to mention, that people should put their questions in the question answer box. And then, of course, remember that you can upvote those that you would most like to see answered. Thank you.
Yeah. Okay, then let's do one more example, but I think we showed you the most figures and different situation. We have shown the potency comparison as well, the sensitivity between strains. So the next one will be slightly competitive, but we'll just do more, which is fine. Again, we did that, yeah. So this is the overview of the analysis you have. Then we click create new analysis. And then we upload the data, choose file, and we go for the fourth matrix mean BB, right? And click upload, yeah? And then firstly check the data to see what data we have. So we have chemicals, a few chemicals here. We have the strain of the animals, we have the TGR as well, tissue, which is only liver. Here we have the exposure route, which is oral all, and then application time. Then we have the dose response SD group size. Then you know again, this is continuous response, but meanwhile it's actually summary because it's reported per dose group. So this is summary continuous endpoint. Then we go further with the analysis. We only do analysis for the 28 days. So let's firstly do some sub-selection. So I have only two. So I just unclick 9 and it's easy. But of course, if you want to go back, you just click all. And the other way is as this, I click 28 if you have a lot of different groups. Yeah, but here is 28. Then give a name, example D. Specify. Firstly, choose the settings. Dose column is dose. Response column is response. We have summary continuous data. And then we choose the standard deviation. It is standard deviation. Group size is this column. And then the covariate, we will use chemical as covariate in this demonstration. And this, it doesn't matter in this case, the BMR, but let's stick to the same. And also we will choose model averaging and just run. That is queued. Now it's in progress. And let's pretend after 1 hour or 2 hours, the analysis is finished. Let's jump to this output. Then again, the most interesting part is this one, report analysis results. Here we get again all the information, a time, input value. Here you see we removed application time and then other settings as well. And then here we have the output of the model averaging for the 3 compounds. And then here as well, we have the plot of the confidence interval of the 3 compounds. And here we can see there are no overlap between the 3. Then in the EA is the most potent one, and we have the MPEEP and then NTTP, the least potent one. And here are the single fit of each model. Just roughly take a look, nothing odd. So the model described the data rather well, and no outliers as far as I can quickly check here as well. And then as usual, here you also get some output of the information for each model of family and which is the best model fit from each model family and got picked in the model error region. And then here you have the model weight. And in this case, we can see actually exponential model describe the model the best, and then the Hugh model and the other two not so well according to the program. The last bit, you can find the constant interval of the three compounds, and then you can continue further with this analysis results. Yeah, so it's a bit similar to the second example, the potency ranking. I think then that's all for the fourth example. And I guess we can continue with the rest of the session.
Yeah, that sounds good. Thank you so much, Guangchao.
Yeah.
We're a bit over time, but we have a break time, and then we have 45 minutes for question answer at the end. So I think we can afford to be a little bit flexible. So maybe we'll answer a few questions now, 2 or 3, and then we can move on to examples of quantal data. One of the questions is, what if the BMD to BMDU ratio, or the interval, is very wide?
Paul, which question you're reading though? I'm ranking the most recent or most upvote?
I looked at most upvotes, but it's changing fairly quickly. So that was one of the popular ones, is asking about what if your BMD confidence interval is very wide?
Yeah, I found it. But this is just by reading. What does it mean? What if the confidence interval very wide? That it is very wide? I don't know exactly what is. You mean like what we need to do?
I guess the person is asking, are there any guidelines? I mean, what if it's 1,000-fold relative to 2-fold? Are there things that you could consider?
Yeah, but I think BMD analysis can have different purposes. In the classic scenario, if for the risks assessment, I can imagine, okay, if it's really wide, that means the BMDL is really low. In some situation, maybe it's unnecessarily low. That means the risk assessment too conservative. That could be the consequences. And if it's not ideal in that situation, then of course you would find ways to improve your BMD analysis. For instance, like we said yesterday, you could consider covariate analysis. If not, you could also consider maybe look at some similar endpoints. But there's always a way to deal with it. But also, like I mentioned yesterday, it doesn't matter the quality of the data, the BMDL stays the same. It's always the dose where the change of the response most likely below the specified BMR. So you can still just use it in either case. If it's 0, then the data is really bad. Then of course you need to figure out some other way. But this is just the idea in the case of risk assessment. But if you are calculating the BMD confidence interval for some other situations, then of course we need to evaluate case by case.
Thanks. Just maybe one more of a comment about nuances for the data file that should be pointed out. I think I mentioned some of this. So when you're naming the variables, you shouldn't use any special characters. So if you want percent micronucleus, don't use the percent symbol. Use whatever you want, PERC or something like that. And then, of course, don't you make your variable names really long. If you do that, I mean, you can do that if you like, but it may interfere with the figures. So it may be difficult for the program to fit the variable names into the figure, the actual diagram. So keep them short, and you obviously want to choose something that's useful for your own reference, but I would say keep them short. And that's what we did in these particular examples. So I see George has his camera on, so we'll switch to George, and then he'll give us some examples, walk through some examples for quantal data, and then time permitting, we'll have a short break. Thank you.
Cool, thanks. Thanks, Paul. Can you quickly just give me a heads up that I am sharing my screen, please?
Yep, we can see it. Cool. It's not in presentation mode yet, but there you go.
Yeah, it's going to be a bit scruffy. I'm going to jump between different formats, so apologies about that, but it is a how-to. And if you were sitting behind my shoulder, this is probably what I would show you. This follows on from what Guangchao and I were talking about yesterday with the Quanto example, the cancer bioassay data. Realize some of you probably wouldn't have been to yesterday's session, and some of you were just dialing in on the recorded version, so I wanted to make this a bit standalone too. So these data that we're looking at are from this EFSA report on nitrosamines, and we're looking in here on these three, NMOR, MPIP, and MPYR, and we're trying to repeat these this analysis, this BMD analysis, using Prost Online to calculate these. So for the example yesterday, we used the EFSA tool online, which uses Prost 70.1, and for this one, I'm using Prost Online at RIVM, which I think as well is Prost 70.1. And just looking at these data sets, I'm going to run through them with you right now. So again, it's going to be a little bit messy, but hopefully I'll get the idea across for you. So going to be looking at this nice risk assessment from EFSA. They, with their risk assessments, with their BMD analyses, they put everything in their reports and in their appendix. And if we go through to their table, I'm showing you this so you can repeat this at home. You can see their benchmark dose modeling side of things just here, the table I've just shown you. And then their decision-making process, why they chose these data. This is all outlined in this dose response section just here. And they went for the original data set for NDMA and NDEA from a PhD thesis, just to highlight that. But we're not looking at that today, but these things are highlighted here. So that's the first part. These are the things we're going to repeat. So if you want to do this in your own time, then look here. Oh, this is annoying. Everything's in the way. In their appendix, they show you exactly what they did. So the BMD analysis. This is quite a useful flowchart we haven't shown today. This is the EFSA flowchart. It's actually outdated a little bit now because they've changed a bit and amended it to include Bayesian. But for today's workshop on quantile data using the model averaging approach, it still suits very well. You just go down this flow chart. If you've got convergence, you see a BMD specialist, and we can discuss this afterwards. Maybe Guangchao could jump in about what the types of thing you could overcome the convergence, how you could do this. And usually it's some kind of problem with the data set. Akaki, so nothing's happening, no dose response over there. We keep going down. With the Akaki AIC, if it's over 2, is there an issue? Talk to a BMD specialist, but we don't really have this issue so much in model averaging because this is all presented really up front. So really this flowchart to me just says, if there's model averaging, go with that. And if you've got problems, you'll see these problems pretty readily when you do the analysis. I better not. Should I get rid of that? I'm just going to minimize it just in case you ask a question on it. So I'm going to go to the data set now. Just looking at this data set, so NMOR, Paul's been mentioning how to run the data. So I just normally put full stops or periods in the place of special characters. Find that's nice and easy. And for quantile data, this is the sort of format you have it in. You've got your dose or concentration, you've got the number of animals with the tumor, and then the number of animals in total just there. Then you save this as a text delimited file, tab delimited file, or you can save it as a CSV file. So that's that bit. If we're going to go for, what's it called? Covariate analysis, same sort of idea we just saw with the continuous data. You just pile these data on top of each other and have another column for the covariate, which is chemical in this scenario. I'm not going to run this analysis, but just show this and then you save it as tab delimited, and then Prost and online Prost will like this. So here is what we've just seen. So this is Prost Online. Again, it uses the same backbone of Prost 70.1 to do the analysis, same as EFSA. You can also download this and put it on your desktop too. So I've just saved it as a tab delimited point file .text. You could do it as a .csv, comma separated file if you wish. Choose file. I'm going to go with NMOR to start with. Upload the data. Everything's nice. You can see the data's been uploaded. There's no problems. If you've added some special characters, this won't upload. If there's some spaces somewhere, if there's an equation, if there's some lettering here, potentially this will show you in this stage, and then you just reformat your data and get it back in. Call this NMOR demo, just in case I've got another one. Same sort of thing we saw with Guangchao, but we're going to look at quantile data this time. So instead of continuous data, where we've got, say, percentage micronuclei or mutant frequency times 10 to the minus 5 or times 10 to the minus 8 if it's ECNGS. We're going to look at quantal data. So we've got our dose, we've got our response, and then we tick quantal. I haven't dealt so much with litter effects, but if there is a toxicology study where during breeding there is a different effect within the litter, then this can be adjusted for within PROST as well, within the BMD approach across all the different platforms. So you'd click on that. That could be alluded to with more detail from Guangchao if we want to later. Here, group size. So this is essential. Sometimes people forget to put this in, then you can't run the analysis. So make sure you put that in. And again, just revisiting this point. So for quantal data, the 10% above the back, the 10% increase in incidence, it's what's used most readily, and sometimes 5%. In this EFSA report, it was 10%, so let's go with that. We saw with Guangchao, he used 50%, and that's for continuous data. And for the gene tox endpoints, we've published a paper, or Andreas has published a paper to show that. For gene tox, and as we stated yesterday, some people might not have been there. 50% for gene tox endpoints is what we're promoting and publishing on based on good analyses. So here we got this quantal data, got your dose, got your response, you got your number, all the data's in there, no dramas at the moment. Model averaging, we'll show you what that means in a minute. For quantal, it's slightly different to continuous. Number of bootstraps, default of 200, and then the AIC criterion of 2 as set by EFSA and all the people that use BMD to adjust for parameters like we've just seen from Guangchao. So this keeps getting in the way. I wasn't predicting that thing at the top getting in the way. So I've run all these already. I thought some of you might start playing around with online PROST and mess up the server. That's what usually happens to me when I run these sorts of things. So I'm just going to run it like that, I think. So we're looking at N more, same thing. That we just saw from Guangchao. We got specifies, that's to change everything. We got the detail analysis. We've got the report analysis. And then if you want to change your plotting at the end. Can I get rid of that? Oh, it's going to go like this. So here, if we go to the details of the analysis, quite similar to if you were going to run this on your computer or normal PROST on R or the GUI version or whatever version you might have on your PC of R. So we've got this and more. Created, expired, remove data. No, we didn't. Quantal, dose, response, number of animals, covariate. We didn't use covariate, but a new addition to quantal data is there is a covariate approach. This followed on from the covariate analysis being developed for continuous data, and now it's available for quantal data too. So that gives us some benefits if we need to use that. And if you look through the EFSA report, they actually did use covariate quantal analysis on there, I think on a couple of chemicals, maybe NDEA, NDMA as well. So it is being used for risk assessment already for quantal data. So that's interesting for you to know. And again, it's quite easy to press the buttons on here and to generate those figures and then interpret them in this sort of way thereafter. BMR, 10% here for the quantal data. Yes, we use model averaging, and here we go. So those of you who've done this previously will see this analysis. You'll see the log likelihoods and the Keiki models coming out, Keiki values. This table you'll probably be quite familiar with. This is probably where you'd stop before model averaging, and you'd use your understanding of number of parameters and the Keiki to ensure which model actually fit the data to the best degree, and then use the BMDs from that thereafter. With the model averaging approach, this is a bit different. It's a bit more advanced. You would then get model weightings for these, so exponential and Hill and two-stage fit the data to a greater degree. They got a high number, and we can see that from here as well, from the Keikis and the number of parameters. Lowest Keikis with a number of parameters being 3. So we can see that just from looking at this table that two-stage, exponential and Hill fit to the greater degree. So they're given better weights. And then these weights are then input thereafter to generate the data and run these 200 bootstraps, which are run. No problems. We haven't got convergence, so we don't trigger talking to an expert, as we see from the EFSA flowchart. Everything's nice. This is all good, all lovely. And we end up with our BMD metrics popping out down the bottom. So that's the sort of PROST type output. The report type output we see from what Guangchao just stated as the real go-to tab. So here you got your output. I've just shown you this one, the details. Now go to the report, and the report would be this one. So you got this. Same sort of idea, just in summary. And now we've got the graphs. So we can see the exponential and the hill fit pretty nicely. The other ones visually look quite nice, but statistically don't fit to as good degree. This is the model averaging output. The dose response fits the data to a great degree through the bootstraps. And then it gives you this summary of the Akaki and the log likelihood and the parameters and the initial BMDs as well. And then it gives you the model averaging metrics thereafter, but not the midpoint, not the BMD 10 itself. So this is it. This is the output. It's quite simple to do, as you've just seen. Some of the analysis can get quite advanced. We can see sometimes this doesn't fit so well. We've got amazing data in this example with nice tight confidence intervals. We've heard what can happen when you've got wider confidence intervals. You have less confidence within that dose response, within that analysis. But what usually happens is you end up with a very, very low BMDL. So if you are going to do a risk assessment on that, it's going to be a very low, overly protective concentration to extrapolate from. So protective-wise, it's good. Precision-wise, not so good in those sorts of scenarios. I'm just going to run through the other ones. So this is what we'd do with, we're going to click on that. If we're going to click on playing around with the advanced plotting. So the advanced plotting, really, if you've got a publication, you want to change these axes. This looks a bit horrible for a publication, having no brackets and all those kind of things. So maybe you want to change that. You can't change all the things you might want to change. There was a discussion about maybe the output from this online post being more suitable for journal publications, and maybe we could talk to that in a bit. And if you use the home version, then you can download the figures in any sort of format you want. So it's a bit better for publications in that respect. But Guangchao might have a little backup for how to do that. So if we're going to look at the next one, that was N more. Now we're going to look at MPYR. Good analysis. The last analysis was loads of doses, loads of animals. This one's fewer doses, but still a high number of animals per dose. So 3-dose plus control with a good study design, a suitable number of animals to detect this dose response to high precision. Still gives you a very accurate analysis and very accurate BMD, as we see by the width of the confidence intervals. Usually people's rule of thumb, or my rule of thumb, it's not written down everywhere, is that anything below, say, 2, or a ratio of, say, 2-fold or 5-fold is excellent precision between the upper bound and the lower bound. Anything below 10 is quite good. Anything below 100 is sort of usable. Anything over 100, ratio between that and that, you can start usually seeing the data is pretty poor. And maybe you want to think about some redoing the analysis, maybe with a covariate approach, maybe going to a different data set if you've got such a data set available from the LASA website, which I'll show you in a sec. So there are different options, realizing you're not going to do another 2-year study to generate more data in most instances. So those covariate type approaches or looking at another data set may be preferable in those instances. But here we see it's a really nice analysis. All the models fit the data in pretty much the identical way. Just a decent dose response, a bit too perfect. So we go to a difficult one. NPIP. We saw yesterday that there are some problems with the top dose, or maybe the top 2 doses of this, making some of the models not fit the data to a great degree. But the Hill and exponential models actually do fit these sort of stranger dose responses to a great degree. And we can still generate BMD metrics, but with a slightly higher ratio between the upper bound and the lower bound. And we see here again that the number of parameters in the Akaki, really the other models aren't fitting the data to a confident degree. So we'll just stick with the Hill and exponential for the model averaging, and that's how it will mostly be calculated. So 0.5 and 0.4 respectively. Ways to improve this analysis. Potentially there's just been some issues in the top doses. I think we saw that they were only 34 animals per dose, and with the other doses, they had high number of animals. There's also a 0 in the control. So there's a few issues with the study design that you can only see thereafter in these types of analyses. So they weren't to know at the beginning, but it is a bit problematic, this one, and quite interesting for today. So I don't want to eat into too much time. I think I've got 5 minutes left, and we can always go back to this, but just show you, if you wanted to do this in your own time with additional data sets, you go to the LASA database. This is the old gold database that was in Berkeley. It now resides on the LASA website. And then you just type in your chemical name. Cas number's usually easier. And then this shows you in pretty similar format, but more interactive format than the gold database, all of the data for your chemical of interest. Then you can pull those data out. They actually have it in a nice format, so you can pull it out in Excel. And then you can just run those data as you wish through this approach, which you now know how to run. So I didn't want to get too advanced with all of this. I just wanted to show you it's not that hard. Get involved. Start looking at these data in your own respect. Realize that this is, if you want to read around the risk assessment side of things, then the EFSA report really highlights why they made all these decisions. What you could do maybe if you wanted to compare it to a TD50, a BMD 50 could be comparable to a TD50. And there's actually a publication coming out from people looking at BMD 50s and BMD 10s comparing to TD 50s and TD 10s from Joel Schlingelman and Larsa and myself on these sorts of comparisons. So there's lots of fun things you can do once you know how to do Prost, and now you do. So I think I'll pass back to the platform chair, which is Paul, and I hope that was all right and not too basic.
Thanks so much, George. We're back on time. We're a little bit early, but that's fine. So in the program, we've allocated 15 minutes for a short discussion period, and then we can go, then we'll have a break, then we can come back for more discussion about any of the things that we've discussed either yesterday or today. So any of the course content. The top one that I see in my list is asking about an example where the models do not fit the data very well, and I think George just showed an example, and asking about what the AIC value would be in cases where some of the models don't fit the data very well.
Yeah, I think the NPIP example sort of highlighted that, as you stated. So the two of the models fit the data very well with, I think it's 4 parameters. The other models were set at 3 parameters, but the AICs were so huge that what it highlighted was that only those 2 models should really be put forward with high weight in the model averaging. And Guangchao's coming in to correct me, I'm sure. Go on, Guangchao.
No, not really correcting. Just one thing actually. For both log likelihood and AIC, it doesn't make much sense just to look at their single number. So it's the comparison between the AICs and log likelihood of different models that gives more useful information. And let's just show you, how well the model fit comparing to the basis. Yeah. So the basis, actually, we did not go too much details into the list. Otherwise, you see actually on top of the list that the full model, there's no model. Without going much details, I'm just saying this full model and no model are actually setting the basis for the worst case and the best case. And then all the other models are actually evaluated based on those. So a single value of those indexes is not really meaningful.
Thanks, Guangchao and George. There's another question about the AIC criterion. Why is it set at 2? And are there instances where you want to change that value?
This 2 is actually just standard in the EFSA guidance. I don't know where did this 2 come from, but it's just a guidance, and then everyone just use it as default. Yeah.
There was an additional comment about the quality of the images. And so that's an interesting comment. I mean, George, you mentioned this a little bit, and specifically images that are required by publications. Is there a way to improve the quality of the images?
Yeah, I was halfway typing and somehow the 2 just interrupted. I think for now, for the web 2, it's a bit difficult. I think I would just check with the IT colleagues if this can be done. But if you use the R version, R standalone version, that's always possible.
Yeah, just to come in on that, when you do use the R version, I find saving it as a PDF is usually lossless and has the best resolution for publications, just from experience.
Yeah, in my own work, I use the R version, and I save it as a meta file, or I copy and paste it as a meta file into a Word document, and that seems to work pretty well. You can stretch it, and it seems to, proportional stretching, and it seems to work quite well.
but most publishers will prefer EPS files, I guess.
It's variable depending on the publisher. Sometimes you can
Yeah,
do
I mean, EPS can be specified as output from the R file, from the R version, so that works nicely.
Right. There's another question about is there any difference in analysis results if you input individual versus summary data? That's an interesting question. Maybe you can address that one, Guangchao. If you have a data set that's individual and you convert it to summary, would you get a different result?
That should be really, if there is difference, it should be really minimal. Most of the time, if the purpose is just to do BMD analysis, I would not imagine that makes a huge differences. But for researching purposes, it's always better to go with the raw data, if it's possible. For instance, just to validate the distribution of the responses, one example.
I think I tried that once to investigate the differences, and there wasn't, if you didn't like insert a rounding error up front. So that's where the differences come from, actually.
I know in my own work, I like Guangchao has mentioned, I tend to use individual animal data if they're available. If we're working on data collected from the literature and we have a mixture of summary and individual, I'll convert it all to summary and then do the comparisons between the different data sets in that way across whatever covariate is of interest. If it was chemical, for example, that's less desirable because there's an extra step in there. You have to convert data to summary, but it's a way around working with data that might be mixed in terms of individual data from some publications and summary data from others.
Yeah, just to add a little bit, maybe a bit too mathematical. I do believe that in most of cases, when the response is reported per group, the summary is actually a arithmetic mean, right? But actually, in PROST, due to the assumption that a continuous data, for instance, has a log normal distribution, that it needs to be converted to the genomic mean. So that makes some differences, but it's really subtle since we talk about this.
I see that Renato Cardoso has two questions. That was one of them about individual versus summary data. And then he's asking about the number of bootstraps. Are there occasions where you might want to change that value, change it from the default?
Very good question, actually. So 200 is actually the default right now in ProstWeb, as well also the IFSA Prost. I also had discussions with my colleagues why there are 200. The idea is actually when the data is quite okay, 200 is most of the time sufficient. So the more the bootstraps, yeah, the more resources it takes, longer time, longer CPUs. But how many bootstraps are sufficient actually really depends on the data quality. So if people really want to be very strict about how many bootstraps are sufficient, then you really need to set different bootstraps until the BMDL and BMDU doesn't change much. If you are not sure, I think I also did analysis with 1,000-ish, but it's very case per case, I would say.
Yeah, I know in my work, when I've tried to use increasing number of bootstraps, as you mentioned, Guangchao, it takes much more time to analyze the data. I think I did an analysis with 7 compounds using model averaging, and it took several hours. So that's something to consider. And I think if you compare, like Guangchao was alluding to, 1,000 versus 200, I don't think you'll see an appreciable difference in the BMDL. Maybe a rounding error, like Andreas mentioned.
Yeah, the thing is, it really depends on the data quality. Like we said, when the data is good, the model knows where to go. Increasing a lot of number of bootstrap doesn't make much sense, but it's always when the data quite poor regarding those response relationship. And that's a bit hard to evaluate just out of my head, yeah.
I'm just sorting through the questions here to look at the popular ones. There was a question about BMD tools and validation of BMD tools, saying that we didn't really understand the question yesterday, indicating that validation of software tools is important for regulatory purposes, and could we elaborate on what's being done to validate the tools? I think you commented on this a bit, Guangchao. Maybe you can elaborate a bit.
Yeah, I do agree. So the validation is quite important. Like I said, so for pros alone, I don't know the other case. There were simulation studied before to check the coverage of the BMD counts interval by simulation studies. For instance, you do 1,000 times, and then you just check how many times the true BMD fall into the BMD constant interval. So that was done, and the conclusion was the 4 models we use for model averaging is sufficient. But unfortunately, it was before my period here, and I don't think I have the information yet. Actually, we've been talking about it lately. But I do agree with this question. Hopefully, we can publish it later as well if it's possible.
I think that the question was rather towards a sort of formal validation procedure of the software as such, and not a more general publication. So yeah, like for regulatory purposes, you really want to have a validation concept and a validation plan, and this is then much of a big deal. And I think we need to discuss about that, the 4 or the 5 of us, and possibly something like that can be achieved. But it's actually not so easy. And yeah, maybe the person who asked the question could. Oh yeah. Oh, so there is a chat message available on that topic. So I see the need for validation. Yes, a formal validation.
There's a question about using Prost in general and asking, are there any specific reasons to use Prost rather than, for example, BMDS or BMD Express? I don't know if someone wants to take that.
Yeah, well, I suppose that goes partly to your confidence in the tool, I guess. Of course, also there's some differences in the methodology as well. For instance, in the BMDS, different models are used for the dose response, which we think might be not very reasonable. It doesn't sound nice this way, but we are busy with writing a paper about this thing. So eventually, I guess it just depends on how much people trust the methodology in the tool. Yeah. As RIVM, I cannot say we are correct, but yeah.
Yeah, and I think it's fair to say that if the criteria are set the same, so if you do identical analyses using the same tools, you should get the same result. I'm less familiar with BMDS, but that's what I would expect.
When the data is good, when the data is good, yes. When the data is less ideal, the quality is less ideal, then you will get maybe sometimes different things from different tools, because there's room to interpret how things should go.
Yeah, so when we looked at this previously and were comparing the tools, there was some harmonization. So Wout Slob, who was developing Prost, was working with the developers of BMDS to harmonize the quantal side of things. So there's some reference on the BMD software that there is some harmonization. And then for the continuous, Prost was generally a couple of steps ahead because it was easier to update things, and then BMDS would catch up thereafter. And there's lots of reference to Prost within the BMDS software along these lines, particularly to say changing from the default of one standard deviation to be able to use the percentage-based approach as well. So it's always seems that Prost has been slightly ahead of it, and then BMDS has come up thereafter, and there has been harmonization efforts between those two based on what I've just stated. And I think for the other approach, the Express one, that's mostly been around gene expression analyses and big data, really, and that's been the main side of things. And previously, we've critiqued it and said, why do you just present BMDs and BMDLs and not BMD upper confidence intervals? And they've adjusted it. So again, I think there's a bit of a lag with that one as well. So that's my understanding. I think Prost sort of leads, and then some of the other ones are slightly behind, but the validation concept is definitely something we should look into and ensure we have a clear story on.
I see a couple of questions about BMR. So asking why we chose a BMR of 50% for continuous data, whereas in the literature you might see 10%, or in the EFSA literature even 5%. and why we've settled on 50% rather than 5%, for example. And I think Andreas and Guangchao, you've both done a lot of work looking at BMRs for continuous in vivo endpoints, and John commented on this as well, if you want to elaborate.
Guangchao, do you want to start, or maybe?
Yeah, sure. First thing first, it's a demonstration, so it's less important. But of course, we go into why 50% BMR. I think Andreas did some study before to see, what is appropriate BMR for in vivo genotoxicity. And Paul and I also been working on something, and there are also some other, I think, evidences or analysis to show that what is appropriate BMR for in vivo genotoxicity. And then the consensus from different evidence or analysis is that actually 50% is quite okay. That's what we mentioned yesterday. So in that case, I just use the 50% today as a demonstration.
Maybe a bit of an add-on to that. So Guangchao's concept of deriving the correct amount of critical effect size is a bit more complex than my proposal. And my proposal was basically to analyze the background noise of the data, since if the increase above the of the concurrent control is within the background noise of the assay, then it makes no sense to limit this below a critical effect size. And then I compared a truckload of data from both genotoxicity studies and also from general tox studies. And the interesting thing is that, at least from our lab, the general tox organ weights are really within this 5% or 10% range of background noise. While for gene tox data, the background noise is much higher. And this is consistent across labs from industry and CROs and wherever. So the 50% really makes sense for in vivo gene tox endpoints, and those 5 or 10% are the default value, are perfectly well suited for organ weights, but certainly not for gene tox endpoints.
See, George has a comment.
Yeah, so I think I looked at a previous assessment from EFSA on a chemical, and within one of the flowcharts they've presented, there is an option to adjust critical effect size based on the endpoint. And that's actually embedded in the R version of PROST as well for that data set in turn. So you can just use that, and it's based on the assumptions, as Guangchao will better explain than me, based on the assumptions of Woutslob's work on the critical effect size theory to do that for each data set in turn. And it is part of the EFSA guidance as well. And we've just done that on a more macro scale across the endpoint with huge data sets and historical data sets to do it for the endpoint in total rather than just a few data sets in turn. And we find this a very robust way of doing things, and we're about to publish a big paper on it.
Yeah, thanks, George. Guangchao mentioned that I've been working with him and George and Andreas on looking at critical effect sizes in more detail for in vivo gene tox endpoints. And we're using Andreas's approach, where we're looking at the background response and the deviations around the mean background response, but we're also looking at the maximum effect size. And we really don't have time to go into the details, but there's a paper by Woutslab that he calls his effect size theory paper, or it's called something like that, effect size theory, where you can scale the benchmark response relative to the maximum response relative to control. So in cases where the maximum response is 1,000-fold, well, you'll need a very different benchmark response than if it's 5-fold. So we don't have time to go into the details, but you can sort of imagine that if your response is only 5-fold, a 50% increase is quite a meaningful jump, rather than if it's 1,000-fold, 50% is tiny relative to the range in response over background that's possible for that particular endpoint. So we've been looking at that in a lot of detail, and we've got a manuscript we're just about to submit, where we provide details for different in vivo gene tox endpoints. Just maybe a last question before we move on to the break. There was a question about mixtures and whether we can analyze data for mixtures of chemicals. Well, the answer to that is that you can definitely do that. And in fact, you can analyze the mixture data in such a way to get relative potency. So you can get relative potency, and when I say relative, I mean relative to a reference compound. Moreover, you can actually look to see if the compounds are additive. So whether the compounds act in an additive way so that the mixture response is determined by the sum of the products, essentially, of the concentration and potency of the individual chemical components in the mixture. And we've actually done work on that, and we're continuing that with pH, polycyclic aromatic hydrocarbon mixtures. But the caveat is that some of the functions can only be carried out in the R version of PROST. They're not available in the web version. So the web tool has some limitations, and we've been using the R version for some of our mixtures analysis. I don't know if anybody else wants to comment on that.
Yes, I know that Julie Sanders and the Science Arno group with Birgit Mertens are working on a similar thing, and we've learned some things from you as well. I'm involved in that project, and yeah, it looks like a really nice way of doing things, BMD, to do mixture analysis. So yeah, it can be done, and in line with what Paul said, Julie's just submitted a paper on this, so some publications emerging this year and early next year on this topic too.
Yeah, we have a couple of publications that oops, I thought I was on mute. We have a couple of manuscripts that we're almost ready to submit on pH mixtures, so people can look for those, where we use the R version of PROST. So I noticed that it's 8:30, and in our program it says break. So maybe we'll go to a short break, 10 or 15 minutes, 15 minutes, I suppose. People can grab a cup of coffee and do whatever they need to do, and then we'll come back for more questions and answers. Okay, welcome back. I hope everyone had a chance to refresh their coffee or tea. I know I did. So I have a fresh cup of coffee. I'll just wait a second to see if Guangchao, Andreas, and George are on the line. Oh, someone said I'm on mute, but I don't think I am.
Back. I can hear you.
You can hear me, okay. So now the final part of the course, we've reserved 45 minutes for questions and discussion about any part of the course. So if people have any questions, just type them in the Q&A box. I suppose if we have lots of time, Guangchao can show his last example, but it's not really necessary.
Let's see how much questions we can finish.
Yeah, okay. 45 minutes, we can do a lot of questions.
Oh,
so
yeah?
as I look at the questions now, I didn't resort them to see which ones had the most votes, but there are some interesting ones. So one question is about models that don't really fit the data and if they can be removed from the model averaging.
Yeah, I got a question yesterday and I could not finish. Don't fit. So the question yesterday was also like for one model, the BMDL is almost 0 or 0. And then the question was, can I remove it? With PROST, it's not possible, but also we don't think it's necessary. I think maybe I can summarize into different things. The first thing is that the idea of model averaging is to take into consideration how well a model describes the data. So when the model fit is bad, then the weight is also really, really low. And if the model fit is good, that is also the weight is higher. So in this way, it's already taking into consideration how well the model fit is. So that's just how the method is. So we don't think it's necessary to exclude the model manually. And the other, just to add on the question yesterday about one model fit and the BMDL is almost 0. This BMD almost 0, it had nothing to do with the confidence interval of the model averaging. Because like we said yesterday, yeah, you fit the model, you get the average model, and then you sample a lot of data, and each of these data sets are going to give you one BMD. And then you get a histogram, you take the 5%. percentile. That's how you get your BMDL from the model averaging. It had nothing to do with the single model fit in the very beginning. Yeah.
Yeah, right. I was going to say the same thing, that the model weights account for those differences in fit quality.
Yeah.
One question. Oh, George, go ahead.
I think there's been appetite for that with quantal data before, but historically and politically and in a regulatory sort of validation, harmonization type approach, those models need to be tested. So I think removing one of those would go against all the guidances for quantal data BMD analysis, and probably hasn't caught up so much for continuous data, but you won't be able to do that for quantal data for sure.
Good point. One question that maybe John can take is asking about pooling data across sub-studies. So John, you published on this. In fact, I showed the example from your publication about pooling micronucleus data across sub-studies so you could more appropriately look at compound chemical potency ranking.
Thanks, Paul. Yeah, I did publish on that. So I think it's, to my mind, it's a useful tool. It leverages those confidence interval plots we looked at yesterday to really give a visual feel for how sub-studies reproduce and when or if they should be combined. And of course, one of the nice things about that is because of the way the BMD approach works, if there are subtle differences in the vehicle control values for each of the subgroups, then that's going to be taken into account when the models are fitted. So it seems like a sensible way to proceed from my perspective.
Yeah, and I would say that I would add to that it should be done with caution, so that if the studies are conducted exactly the same way, then of course you can investigate whether sub-study has a significant impact on the result. But if there's a difference, for example, in exposure regime, for example, and we've seen that before, then you may want to be cautious, and certainly be cautious if you're working with data from different sources. So if it comes from the literature and it's data from different publications, then you have to take that into account as well. Another question. Sorry, go ahead.
No, I think that's a really good point, Paul. I think your comment on exposure duration is worth just mentioning specifically. I see that catch people out sometimes. We're used to talking about point of departure metrics in terms of, perhaps daily dose, mg per kg per body weight day. But obviously, sometimes exposure durations are not always, historically 28.1 or something, depending on when they were done, when they were performed, relative to when a test guideline became available. So it's worth keeping a really close eye on that one, because obviously 28 days of dosing, for example, is going to have a very big impact versus 14 or a shorter period. So definitely keep an eye on it. Thanks.
Yeah, and I think a good example too is, where I guess you could say it's different sub-studies. In Guangchao's example, where in that paper by Zhao et al., where they looked at 30-day sampling time and 90-day sampling time. So it's different groups of animals, but I guess you could say it's essentially the same study with different animal groups tissue sampled at different times after the final dose. In that case, it was actually only one single dose. There's another question about the R version of PROST and the web version of PROST, and whether there's any fundamental differences between those two versions.
No, not really. It's the same thing. It's all based on the same R package, but just with the R package, there are more functions available, and with the web version, only a few essential functions are activated. Yeah.
And we mentioned mixtures analysis, so that's only available in the R version. And actually, I could have mentioned that one of my staff who's actually online, Hannah Batayan, has been doing a lot of work with mixtures analysis, and it gets a little bit complicated. And if people have issues and they're interested in doing mixtures analyses, they can simply contact us, contact my lab or contact George's, and we can, try and help you out. We did have one question about nomenclature and about the use of BMD versus BMC, and asking for an inhalation study, should it be BMD or BMC? Well, my answer to that would be, if you only know the concentration in the exposure medium and not the dose, then the appropriate term would be BMC. And that's what would be used for in vitro as well. I don't know if anybody else wants to comment on that.
Yeah, indeed. I mean, BMD is just the way we always say it, dose and response, but it can also be concentration and response. as long as you know what exactly that is. Yeah.
Maybe on that point, according to my experience, the data are getting much better if you're using actual TK exposures or PK data instead of doses. Yeah.
Okay. There was one question about the reporting in the results output, that only the BMD confidence interval is given at the bottom of the output, and basically asking where's the BMD value.
I explained that one before. Anyone want to answer instead? Yeah, well, two things. The first thing is the BMD is actually just a point estimate, so it's very uncertain. And we said, the uncertainty of the BMD is reflected by the BMD confidence interval. And in the case of risk assessment, we take the BMDL. So we think actually reporting the BMD is not really meaningful in this way. It can be also misleading. So our question would be, why would you need the BMD in this case?
Yeah, the point of departure for, let's say, regulatory decision making would be the BMDL. I guess related to that, there's a question about use of the BMD approach in for drug during drug development, and someone has asked, basically saying that they have to use very specific guidelines, and are there examples where the use of the BMD approach for interpretation of genetic toxicity data has been submitted for regulatory review? That I did defer to others on that. Maybe Andreas can take that question.
Yes, we have submitted micronucleus in vivo data that have been generated or interpreted with BMD software for a drug development project, and the data were submitted in like 2017 or 2016, and there were some questions back from regulatory authorities, but we could answer all of them, and so that was accepted. I have to add that we also added mechanistic investigational data. The drug in this context was an antigen, and so it's not only based. I mean, the regulatory submission was not only consisting of BMD data, but a ton of other data as well. And Renato, I will send you the literature reference via email.
Thank you, Andreas. Any other comments on that one? George, you've been involved in that a bit.
Yeah, so well, obviously a lot of these things are behind confidentiality walls, but I'm aware of some of these data in line with what Andreas was saying, allowing the preclinical package to be accepted and allowing the drugs to go to clinical trials. And I'm aware of a few examples with that with some colleagues I've discussed this with. And then I know loads that have been submitted and are currently on regulatory people's desks for nitrosamines as well, with the accepted part of that sentence not quite being there. But yeah, definitely some examples for clinical trials definitely being accepted, and then some other examples being harder to get at the current time. But hopefully that changes with our deeper understanding of what we were talking about today.
There's a couple of questions about the output. So one question is that if you're doing detailed analysis of chemical potency, so using the covariate approach, is it possible to get a downloadable table summary of the benchmark doses and their confidence limits? In my experience, you don't get that, and you have to, if you want to present it in Word, for example, you have to type in the values into a table. But I don't know, Guangchao, you have a comment?
No, that's true. Yeah. I mean, I always love this kind of feedback. That's indeed, it's not possible at this moment, and that's just how the tool is programmed at this moment. But of course, I think that's a good point to improve. I do remember we had this kind of feedback maybe a couple months ago, which we do not have time, but that's a good point, and we will take into consideration and put on the list for sure.
Thanks. There's a couple of other questions about poor model fits and how that's handled in model averaging. I think we addressed that, and that the weighting actually addresses that effectively. Let me see about the other questions. One is asking about the necessity for data transformation. Are there instances where it's necessary to transform the data in some way before the data are imported into PROST?
What does it mean exactly? Transform the data in the sense of data format or the data itself?
The data itself.
Yeah.
But then how would you like to transform the data? I mean, our idea is just use the raw data and the raw dose response data.
Right.
Guangchao, I think the question is, would it ever make sense, for example, to take a square root transform of the response data prior to upload to PROST? From my perspective, the answer is no, but could you just elaborate as you're the technical expert?
Oh, we're talking about this kind of transformation. So there's we said there's continuous endpoint, toxicity endpoint, and quantal. I think here this topic only valid for continuous, and for continuous, indeed, we think no. We think actually in the majority of the cases we see that log normal distribution is valid, and also based on theoretical argument, we do think that's actually suitable distribution. So we don't see any need why it would need to be transformed. Only if it's actually defined, strictly defined that way, whatever the situation is, then that could be a situation you consider that. I vaguely remember in the R function that might be something similar, but of course I need to check. If people really need to know the details of this part, you guys can contact me for that. That might be one option. Got the square or got the normal distribution even. Yeah, in R version.
Cool.
And there are, sorry, go ahead, John.
Very briefly. In the R package, you can produce a quantile plot as one of the sub options to see. Don't forget, if you're working with continuous data, then the correct assumption is log normality, not normality on original scales, because those response data on continuous scales are multiplicative, not additive. So there's that option. Guangchao, I don't know if it's possible in future for QQ plots to be available by the web tool, but it'd be pretty cool if that was the case. Maybe residuals too. Thanks.
Good idea, indeed.
There are several other interesting questions, some of which we've discussed earlier in some detail. Someone is asking me about what if you have a common set of controls? So you're looking at several compounds, for example, and you only have one set of controls for that particular study. How would this be handled, or how could this be handled?
Perfect timing. We got exactly the same question from Andreas, I think, a couple months ago. We had an intensive discussion. I still need to search for a final answer too, but two ways. One way is, for instance, one control group, and then 3 compounds, for instance. Each compound has a few dose groups. One way is that then you can imagine we have 4 subgroups. Yeah, control is one subgroup, 3 compounds one subgroup, and in that case, you just tell the tool that we have a common control, and the other compound will be just borrowing the information on the control from that control group as a single subgroup. That's one way. And at that time, George also mentioned, okay, how about I randomly assign that control group to either of the compounds. So 3 compounds, 1 got the control group, the other 2 doesn't, and still we do the covariate. That's another possible way. I did the comparison, and then they are kind of like comparable. I did not get the chance to talk further, but those are 2 possibilities I could think of at this moment. Okay, John.
Guangchao, that's really fascinating. I just wondered if I could ask a follow-up. Presumably, what you would not recommend is duplicating those controls multiple times. Could you just elucidate to that? Because I've seen people do that. Thanks.
Oh, yes. That's not recommended. The reason is if you duplicate the control, you're actually assuming that for the other compound, your control is exactly the same as the only actual control. And in that case, your analysis will be overly optimistic because you're using the same information and you assume like that also applies for other information. But in the reality, for instance, if you had 3 more controls, you can imagine actually their variation will be different. So the whole spreading of the data will be different. So that is not recommended.
Do you see a possibility to include the one control group compared to several substances with different treated groups feature to be added in a future version of PROST?
I don't see why that need to be added as a feature, because this can be easily done in your data formatting. You can either put the control as one compound, or you can just say this is one subgroup itself.
Right. I see a question about benchmark response for error-corrected sequencing data, mutation data. That's a really interesting question. So Guangchao and Andreas and I and George, we've looked at PIG-A as an endpoint, transgenic rodent mutation data. Wout's lab has looked at micronucleus, and I know Andreas has looked at COMET. To date, nobody has looked at error-corrected sequencing data in detail. I'm happy to report that with Francesco Marchetti, we've hired someone to collect all the individual animal dose response data for any next generation sequencing study that's looked at, used that approach to determine mutation frequency. And that work is underway. So we're collecting all the data now, but we'll do detailed analysis of the data to determine what would be best for a benchmark response for that endpoint. But the jury's still out on that, as we would say in English. But we'll certainly be looking at that in great detail in the near future. George.
I think John's hand was waving before me. Go, John.
Oh, go ahead, John.
Well, thanks, George. Just to say, with the error-corrected sequencing, I think one of the reasons why this is complicated is you can imagine the ability to detect a mutation is somewhat dependent on, it's entirely dependent on the panel you choose. And it's also, so at the same time, it's also dependent on the read depth. So you've effectively got read depth and length, which influence the probability of whether or not you have enough sampling to detect a mutation. So basically, it makes error-corrected sequencing quite complicated because it's not one thing in terms of the response data that's generated. So really excited to see what you do in that space, Paul, because there's certainly lots of questions there and certainly very relevant to what we're doing in-house at GSK. Thanks.
Yeah, maybe we could communicate on that, because hopefully we'll do it in a robust way that will be useful to anybody who's working with those data. I see another question about, it's more of a mutagenicity question from David Schuster, asking about what response value we should use if we have information about mutation spectrum. So that's an interesting question, and it relates to error-corrected next-generation sequencing data. If we have information on total mutation frequency, and let's say whatever specific type of base pair substitution mutation, which one should we use in our dose response analysis? I don't think there's really an answer to that question. It's whatever fits your research question. I don't know if anybody has any additional input.
Just come in quickly, respond to the last question as well a little bit. So some people are already doing this, and they're already using total mutation frequency for all of the different sequences and doing BMD analysis on that, using 50% critical effect size, and the BMDs they're generating are below that from the same, sometimes the same animals, but using the TGR endpoint. So even at the current time, we're seeing with those two assumptions being made, it's still producing protective BMDs compared to the gold standard of TGR. So just putting that out there that people are already doing these two things, using total, using critical effect size of CES, and it's looking lower or comparable to TGR. Sorry, John, for jumping in. Thank you.
No, that was perfect, George. I think also, David, it maybe depends on what you're trying to do, like whether you're trying to get a point of departure and compare what that is relative to what you might get from TGR, or whether it's to get some, quantitative potency estimate that you're comparing across different mutation spectra, for instance. In the latter case, it shouldn't matter so much, and indeed, it might be argued that choosing a slightly larger benchmark response size makes sense because it's going to be more. You're going to be interpolating your data more within the region of the responses coming away from the background. So you can imagine that might give slightly thinner confidence intervals to enable your comparisons. Thanks.
There's a question about model averaging and why we need to use the bootstrap approach. Why do we do that?
Why? But I think bootstrap is just one part of the model averaging, and then how should I start? So the idea of model averaging is that different model may give you different fit to the data, of course, and then we would like to take this into consideration. And then the bootstrap is just to theoretically sample different data set based on the experiment data. And then in that case, you get a model average BMD confidence interval. And the idea of bootstrap is that you theoretically consider that you are doing the actual experiment multiple times. But by actually doing it in reality, you do it with simulated data set. And then you would like to see, how much is the spreading of the BMD, and then you get the confidence interval. So if you're only interested in the bootstrapping itself again, the concept of bootstrapping is just theoretically performing the identical experiment, because every time we do the experiments, we're actually sampling from the infinite population. And then each time you can imagine there's some maybe differences of your results. And the idea of bootstrap is just to take this sampling error into consideration. I'm not sure if that's understandable or not.
I think that's an effective answer. There's a question about model weights and whether there's a minimum weight that needs to be respected. I think you sort of answered this before, Guangchao.
Yeah, there are no minimal. I think could be almost zero. It's just all the model weight, they sum up to 1. That's it.
Is it okay to add something to that briefly, Paul?
Of course.
I see a lot of questions coming in on model averaging and weighting. And Guangchao has said what I'm about to say before, so apologies for the repeat. But you can imagine part of the issue would be we've got all these different models, and which do we choose in order to have our BMD metrics? And that's a really knotty problem in effect. And that's the thing that we're divorcing ourselves from with model averaging. We then just have the weights. They may be absolutely tiny and virtually no information is being contributed forward, but it divorces us from this problem of having to choose and defend why we've picked a particular model. And I think that's a really critical thing. It allows that process to much more fluidly run on rails without worrying about all of those technical decisions.
Yeah, maybe add to one thing about concept. So the whole point to move from one single model fit to a set model fit and use model averaging is that we are not interested in the best model fit, because when we perform experiments, there are always sampling error. Like we said, if you measure people's height, if you get people from different regions, you may get a good reflex. But if you only sample from certain regions, maybe it could be quite different from the reality. That's the idea of sampling error. But meanwhile, we also have experimental errors. People do measurement, nothing can be perfectly randomized or other things. So that being said, the best model fit of the data does not mean that fit is closest to the reality, like we mentioned yesterday. So maybe the second best model fit is closer to the reality than the first one, but then we wouldn't know because we don't have the data from the infinite population. So this whole idea boils down to that, let's just look at all the models and look at all the possibilities, and then we get a result based on that. So that's the whole argument of that.
There's a question about alternative potency metrics, I guess you could call them, and how the TD50 that's used for carcinogenicity data interpretation, how that compares with the benchmark dose. I don't know if someone wants to tackle that.
I'm happy to speak to that. George and I currently got a paper that's submitted but not yet available that looks exactly at this and compares BMD outputs with a large number of TD50 values for a big set of compounds. So keep an eye out for that. In effect, the TD model is fundamentally more simple and doesn't necessarily have the ability to describe all forms of dose response data as well as the benchmark dose model, but it is something that's embedded traditionally in cancer risk assessment. Effectively, the BMD models and using a family of models should give us a much better ability to reflect the uncertainty in the experimental data than the TD50 approach. Thanks.
Yeah, just on that as well. So traditionally, just the TD50s itself are presented and discussed and used for risk assessment with no discussion or presentation of the confidence intervals around it. So you don't really know the precision of that risk assessment metric, whether it's from absolutely rubbish data or from perfect data. And in that way, it's not as protective as the BMD that does have that measure of precision. And in a bad data set would have a lower BMDL and protect the human population, where a single dose metric wouldn't have that level of protection. And also that talks to one of the other questions as well, how it compares to all these approaches compare with poor data. The BMD remains protective, and the TD50, I think, would suffer more being a single dose metric approach. I think Guangchao's looking quizzically at me, maybe to expand.
No, nothing.
We have more questions about model averaging, but before that, I think there's an interesting question about historical controls, and if it's possible or if it's desirable to use historical control data from a particular institution as opposed to controls from the same study, so concurrent controls versus historical controls. I don't know if anybody wants to address that. Not necessarily a BMD analysis question, it's more of a general dose response analysis question.
But I think we did have this discussion together with Andreas as well, right? About using a historical control data just to, better comply with the 3R principle. But back then, I think we said, the idea of the control group is actually to set a threshold to the other dose group, how much change of the response according to certain doses, certain exposures. So that's the best idea. And of course, in that situation, you would only want those to be the major or the only cause of the change of the response. So basically, and in the experimental settings, we try to randomize the animals, consider also the caging, whatever, so that we can even out the influence of the other factors as well. But I think it's still kind of apply for the use of historical controls, because you would not use, for instance, a control from the rat to the mouse, because that two different strains. But also you would not use data from the old mouse with doses which tested on the young mouse. It's the same idea.
I mean, there has been an initiative on virtual control groups going on since years, and this has been published recently, and it's way more complex than we could explain in this short time. So I would say the answer to that question is it depends. But in general, I would certainly advocate for the use of virtual control groups, or for properly validated methods like that, let alone from a 3R point of view. And it certainly depends on the endpoint. I mean, according to my experience, for the micronucleus in vivo, the data are so stable over years. You will have exactly the same background level and noise in the controls, while for example, for Comet, it's a whole different situation. So it's not so easy to answer that question.
I see George and John have comments.
I think I got quite a simple brain, so I like to think of it in simple ways. And I think that we still stay with this tier 1 use historical controls and pairwise testing and trend test for that first yes/no decision-making step. And then we're really talking about the second step, as when it's positive, what do you do with it? How do you measure potency and points of departure? And then we come in. So still embrace previous ways of deciding, and then we build on top of that is my simple way of thinking about this. Sorry, John.
I think one thing that sometimes gets lost in the historical control discussion is when we make a measurement, there's always an error term, and it's to an extent unique to the day in which we're making the measurement. And hopefully the error term is small and consistent, but that's not necessarily the case. And it's always there and present in those vehicle control estimates. So that's something that we need to think carefully about.
Thanks, John. We're running low on questions, actually, which is a good sign. There are several that are repeats, but a couple that we haven't addressed. There are a couple of comments. So one comment on validation saying that similar for QSAR models, the regulators need to have confidence in the output so that validation is essential. There's another comment. There's a comment about model selection and asking why, well, I'm paraphrasing here, why isn't a linear model one of the options or one of the models selected for the model averaging approach? I don't know if you want to get that gets to be it's as we would say in English, it's opening up a pretty big can of worms, but maybe you want to take that one, Guangchao.
For linear, well, I think maybe the participant meant actually at the lower dose range, maybe because you can never imagine dose response will be linear all the way from low to high dose range. And then as that idea, firstly, I would like to ask actually what is actually the evidence that it is linear, exactly linear at the lower dose range. If there are studies or proofs or analysis show this, I'm very interested to see. Meanwhile, actually the model we included, it does not exclude the possibility is more or less linear at the lower dose range. That's just the flexibility of the model. I'm not sure, I cannot hear any feedback, but that's how I understand the situation.
Okay, I'm just looking through the questions here. One question about, if I'm interpreting it correctly, about the ability to do covariate analysis in BMDS. I haven't done this myself, but if anybody knows about this.
I haven't looked at the updated versions. The older versions, it was a definite no, but hopefully they've caught up. I don't know.
I'm not familiar with the BMDS in that way.
Oh, Guangchao, you're saying you still have 23 questions to be answered? Well, I'm just sort of sorting them by upvote. So yeah, I'm only seeing part of them that are at the top of the list. But here's one that I didn't see until now. How does parameter C influence the analysis, especially if there's no evidence of a plateau in the data?
If that's the case, then the model would not include the C in the analysis. So basically, the model itself, at least for pros, I will check. Yeah, the model we see without C. So you also see in the output later. I did not go into details. If actually the model without the C, without the plateau, gives a better fit than the one with, then of course, then the one without will be the best model fit. So the two will check this aspect.
Thanks. There's a question about converged, and what does converged equal 1 in the output mean?
Oh,
I asked the same question last week.
Yeah. Do you want to try, Paul?
No, you go ahead.
So converged equals 1 means the fit is converged. So to put it in a simple way, what is converged? Fit is converged. It means actually the model fit. The best estimate of the parameters are found in the analysis. So when you have a data, you want to analyze it, you want to estimate model parameters. The tool does the way that every time increase a little bit of the change a little bit of the model parameters, and then you have a fit, and then you change it again, you have a model fit. So each of these changes is like each iterations. So when the change of the model parameters do not actually influence much anymore the model fit, so that says that you find the best parameter estimate based on the data, and that is the situation when you call the fit is converged. Yeah, and if it's not converged, I think it would just a false, and then something wrong with the data.
Right.
Yeah.
One question about regulatory submissions, and is there any advantage to using Prost Online or the R version of Prost? Are there any differences? And a very easy answer to that question is we've addressed that a little bit. There are no fundamental differences between the R version and the web version, except to say that the R version is a bit more comprehensive. So there are functions in the R version that are not available in the web tool. But essentially for the routine dose response analysis, such as we've demonstrated, there shouldn't be any difference between doing it in the web tool versus doing it in the R tool. Does that seem reasonable, Guangchao or John?
It is, indeed. One thing I do want to mention, not to help us or not help us. So when you do your own analysis with the R version, you are doing everything on your own computer or laptop. All the information is stored locally. When you use the web tool, you need to upload your data into the web tool. So there is a discussion among my colleagues and I about how to handle those data regarding confidentiality. So I would like to just remind you that this is something, and we're trying to figure out what's the best way to handle it. And of course, and then in the future to improve this confidentiality and security as well. But this is something that you should keep in mind. Yeah.
Go ahead, George. And John, well, John has a comment.
I'll be quick. Guangchao, presumably it may just be that the version of Prost that's available for download to use as an R package is slightly different than the version number being used as the back end of Prost Web. I think 0.9 versus 71.1 at the moment, for instance.
Yes. So the R version, R package is uploaded regularly when it's needed, and the one on the web tool, they need to be implemented. So that takes some extra work, and we are depending on the other colleagues, IT colleagues as well. So I'll finish with saying IFSAT also has a Prost web version, which is the IFSAT BMD, small letters. If any security reasons is the concern, then that can be another option as well. It's also Prost.
Okay. Yes, I think in practical terms, it's a bit like the GUI, the graphical user interface version of the R package. Whereas if you use the command line version of the R package, it can get quite complicated for a new user with quite a lot of other options. So if you're submitting regulatory submission, someone else trying to repeat it might be using the R version. You could go down the wrong track and generate different BMDs based on a faulty analysis. So it's more prone to error using the R code version if you're not so used to BMD is my advice. So I think going with the simpler online or GUI version, there's less decisions to be made, is my experience with it.
One question that's more about, I guess, regulatory interpretation of genetic toxicity dose response data. So basically saying that if I want to establish a POD from in vivo mutagenicity or carcinogenicity studies, what if the studies have not been designed in such a way that BMD modeling is useful? So few doses plus control, maybe only 2 doses plus control. And are there instances where you might default back to the NOEL instead of using the BMD? Anybody want to try that one?
I would say just just think out loud. 2 doses plus control, 3 doses plus control. You can still try the BMD. You never know the quality of the data until you do the analysis. Only if you say I have only 1 dose, then I don't think BMD is meaningful. But otherwise, yeah, just try the BMD instead.
Yeah, and I might add that in the analysis that Guangchao and George and Andreas and I have done, looking at the critical effect size or benchmark response for in vivo endpoints, we have a lot of data sets that are only 2 doses plus control. And in many cases, they work just fine in using the BMD approach. So like Guangchao is saying, it depends on the quality of the data. So it's case-by-case basis.
And with the NOEL, you usually want a dose or two below the no observed effect level. So you definitely wouldn't get that with 2 dose plus control. So definitely not the NOEL, but possibly the TD50. But then you would have you get a TD50, but with poor data, and you wouldn't have the measure of precision, as we said earlier, with the confidence interval. So the BMD gives you that information that it might not be the best analysis, but it's analysis you can still discuss.
Well, we're getting near the end of our time period for questions and answers. I was just going to add, related to that question about regulatory decision making, and I mentioned this yesterday, that people may notice that we haven't really talked about risk assessment and the use of BMD values for determining a point of departure that's used for regulatory decision making, and that was quite deliberate on our part. So that's a very separate topic, and it would probably need another day or another morning to actually discuss that in any detail. And that it becomes quite a controversial topic, and we'll have to think about, there was quite a lot of work to put this course together, but maybe we'll have to think about a follow-up course that's dedicated to risk assessment specifically and regulatory decision making. So there are more questions in the question answer box, but they tend to be repeats of what we've already addressed. One question that we could address that we've already addressed in some part, to some extent, is where do the simulated data sets for the bootstrap come from? I think you've already sort of addressed this, Guangchao. Maybe just elaborate a bit.
Yeah, so this simulated data in the bootstrap, they come from the averaged model, which is based on the fitting of the experimental model. So if we go back to the slides yesterday, I mentioned, firstly, we have an experimental data set we upload, and then we use 4 model families, fit them, and then we pick the best model of each family, which means we have 4 model fit, and then each of these models gonna have a weight, and then we just do the we get the average response based on the weight, and based on the model, each model, and then we have an average model, and then of course the variation how we sample the data set also depends on the experimental data, and then based on the variation as well as the average model, we will sample the data set as many times as specified by the number of bootstraps. Is that a clear answer?
Well, it's clear to me. Hopefully it's a clear answer. I see that we're at 9:30 now in my time zone, which brings us to the end of the course. There are still more questions, but they're variations on the same theme. So I think we'll just stop there. I just wanted to thank everyone for participating in the course. I hope people learned a lot. And I wanted to thank HESI GTTC, or HESI, the parent organization, for hosting this webinar activity. And thank my co-chairs, Guangchao, George, John, and Andreas, for helping develop the course and for teaching the different portions of the course. So thanks so much for that. And I guess we'll end there, and people can move on to their next meeting. I see we're getting a lot of thank yous in the chat box.
Just to add, Paul, I think people can contact me at least freely. The RIVM Pro's website and the email just there, whatever question they have, if they would like to know more.
Yeah, I think if there's actually an email address for PROS, a help email address, and I think when you send emails to that address, they go to Guangchao. Is that correct, Guangchao?
I'm the people taking care of the daily basis. Yeah, it comes to me. Yeah, so now you know the face.
And I know in our own case, in my lab, we've had situations where we needed some help about some details in using PROS in R, and Guangchao was very helpful.
Thanks. Yeah, doing my job.
Okay, thanks so much, everyone.
Thank you.
Bye-bye.
Thank you. Bye.